Welcome back to a new edition, The Science Behind the Coronavirus. In this fourth installment of this series, we're looking at a very important question. The viral variants that are circulating worldwide in the effort to protect against them. Joining me today for this discussion is Professor Shabir Madhi, a virologist who has led South Africa's clinical trials of the AstraZeneca coronavirus vaccine. Days ago, the South African government made a stunning announcement. It would halt distribution of 1 million doses of the vaccine after tests found it didn't prevent disease from the variant that's ravaging that country. Professor Shabir Madhi has led this investigation in these vaccine trials and delivered these stunning results of his research during a government briefing.
When looking at vaccine efficacy against this particular variant, we have not been able to determine that the vaccine actually protects against mild to moderate illness.
Today, we'll discuss what this means for South Africa and for the future of vaccine development and distribution around the world. We'll also hear directly from Professor Madhi about the enormous sacrifice researchers are making in order to determine how best to protect the population of South Africa against the virus and this new variant in particular. Before we start, as full disclosure, my company ImmunityBio is developing a COVID-19 vaccine, and we've been given permission to begin trials in South Africa. We are going into the heart of the storm and with our vaccine trying to address this variant. So now I'm pleased to have Professor Shabir Madhi join me from Johannesburg. Professor, welcome.
Good day. Thank you for having me.
So let's start a little bit about the really important news you released on the webinar and the paper that you've now published with regard to what you found with regard to this variant, and maybe for our lay public. And this specifically affects not only the South Africans, but the spread of this variant throughout the world. If you could just very briefly describe what you found.
So, Patrick, the study which we did was a phase 2 study of the AstraZeneca vaccine. Because it was a phase 2 study, we were somewhat guarded in terms of the spectrum of individuals that were allowed to be enrolled into the study. Really between 18 and 65 years of age, without too much in the way of underlying illnesses that could predispose them to developing severe disease. The main objectives of the study were foremost to establish the safety of the vaccine, and secondly, to determine whether the vaccine would protect against COVID-19, irrespective of severity of illness. The summary of it is that after having received 2 doses of the vaccine, the final conclusions of the study was that this particular vaccine did not protect against mild and moderate infection due to the SARS-CoV-2 that was a consequence of infection with what is known as a B1351 variant, which is the variant that evolved in South Africa and has subsequently spread to many, many other countries, including the United States. What is important of this variant is that the virus has developed mutations, which makes it resistant to the antibody that is induced by past infection with the original virus that was circulating, and seemingly also resistant to the antibody that is induced with the AstraZeneca vaccine, as well as probably many of the other first-generation COVID-19 vaccines that are being deployed throughout the world. But at the same time, I think it's important to emphasize that that doesn't necessarily mean that this particular vaccine or any of the other vaccines won't protect against severe disease. And what it tells us is that to be able to protect against infection probably is a much higher bar than what is required of a vaccine to be able to protect against severe disease.
When you first saw the results of those 2 figures of this neutralization, it's a devastating, actually disappointing finding. You'd hope not to see that. How did you feel? I just want to get your personal experience about that. How did you feel about that, Professor?
Yeah, so the lab results came in about a week before we unblinded the study to see what was the clinical effect of the vaccine itself. When the lab results came out, it wasn't completely surprising because it was earlier work that was done by Alex Siegel as well as Penny Moore against what was in convalescent sera. And when we saw the results for the AstraZeneca vaccine in terms of the reduction of the activity of the antibody against the virus, that was obviously disappointing. And it sort of made me a bit pessimistic in terms of what to expect of the eventual efficacy readout once we unblinded the study. In terms of what has subsequently happened, within a few days later, we were ready to unblind the study because we had accrued sufficient number of COVID-19 cases to get an answer to the question that we posed. And the question that we had posed was whether this vaccine would have at least 60% efficacy against all severity COVID-19. And I recall walking out of my office, about to go home, and down the corridor came the statistician with a very tearful look on her face. And then she told me, listen, I've done the analysis and pretty much we don't have any vaccine efficacy to speak about, which was obviously extremely disheartening. But I think after it had settled in, I think that I realized just how important this finding was to the field of COVID-19 vaccines. It was hugely disappointing to the rest of my team, to the number of people that have worked on these studies. I mean, across South Africa, there's 7 teams and probably about a quarter of the staff that have worked on these vaccine trials in different settings were actually infected with the virus during the course of conducting this trial. So it's been a huge amount of effort, and it has come at huge costs to the staff that have been working on these studies. So obviously, everyone has been disappointed. But at the same time, we know that we've now contributed to the knowledge set, which tells us that the game is far from over when it comes to developing a COVID-19 vaccine that is going to be resilient and that's going to be able to actually eventually gain control of the transmission of this virus.
You thought that, or you made a statement that this virus will be here in our lifetime. And how long will we be dealing with this?
Yes, I think COVID-19 is here to stay. The virus itself is not going to be eradicated anytime soon, extremely unlikely. But I think the target right now is to try to reduce the frequency of these resurgences and the magnitude of the resurgences. And that is what the focus needs to be. It's not about eradicating the virus. Unless you're an island nation such as New Zealand, or Australia, or Iceland, and you pretty much keep your borders shut or have an extremely strict quarantine process when there are incoming visitors, it's highly unlikely that anywhere else in the world you're going to be really successful in eradicating the virus. So we probably need to recalibrate in terms of our expectations of how to manage the pandemic, as well as in terms of what to expect of vaccines. So what probably would end up happening, for all intents and purposes, is that this will probably become the next common cold, one of the other coronaviruses that becomes almost endemic in humans. We've got 4 other common cold coronaviruses that cause more of a nuisance than anything else. But I think in the instance of this particular virus, we probably will still see ongoing number of people dying from COVID-19 into the foreseeable future with or without vaccines. But obviously vaccines are going to tremendously reduce the number of deaths that will result from this infection with this particular virus. And in addition to that, it would relieve the pressure on our healthcare systems. So vaccines are probably the only sustainable option that we've got at hand to be able to bring the pandemic under control, to reduce the frequency of the resurgences and to reduce the magnitude of those resurgences.
Give me a little bit your understanding of how this mutation arose. And it's not just in your country, and I say our country because I was born in South Africa, but it's going to rise elsewhere. So if you can just give us your insight and your experience, starting from Eastern Cape and all the way up South Africa and all the way to Wits, and how rapidly it spread.
So I think you're absolutely spot on. Some of the mutations that do take place in viruses as well as other organisms are pretty much an error of the reading code, which codes for the different proteins. And that error can take place at random. But more importantly, the type of mutations that we're experiencing are a consequence of the virus coming under what we call immune pressure. That is, the virus is being exposed to these antibodies that are either induced by natural infection or through vaccination. And for the virus to continue surviving, it needs to adapt itself to evade those sort of antibodies that are directed against it. What happened in South Africa is during the course of the first wave, and similarly probably in Manaus in Brazil, during the course of the first wave, there was a tremendous force of infection taking place. All of the sero surveys, when we test blood samples from individuals to see if they've been previously infected or not, All of the serosurveys that were conducted in South Africa indicate that as many as 30% of South Africans were likely infected during the course of the first wave. So that means there was a huge number of people that had developed antibody against the original virus that was circulating. Unfortunately, the virus continued circulating in South Africa even when that first wave started to plateau off. And consequently, with this huge amount of ongoing transmission of the virus, which was now faced by these antibodies that was being induced, for the virus to continue surviving, it required it to mutate, to try to. And essentially, these mutations are a consequence of what we call immune evasion, which is where the virus are adapting to evade immune antibodies specifically that's been induced by natural infection in our instance. But as you correctly point out, as vaccines, COVID-19 vaccines are rolled out, and especially if the rollout is very sluggish, in that you've got a very slow uptake of those vaccines, and if the virus continues circulating, it provides almost fertile ground for the virus to mutate against those antibodies that have been induced by vaccination, let alone by past natural infection. So this is something which is a phenomenon of not just the SARS-CoV-2 virus, but something that exists for influenza viruses as well, as an example. And that being one of the reasons why each year we're needing to revaccinate against influenza virus, because of what we call as antigenic shift that takes place, or antigenic drift that takes place, in that there are the subtle mutations that allows the virus to start evading the immune responses that's been induced previously against it. So very much what you said, basically is there's more antibody around directed against the virus. For the virus to continue surviving, it needs to adapt, it mutates to try to evade that antibody that's been directed against it.
I understand that South Africa has now put on hold the 1 million doses that AstraZeneca has shipped in from India for distribution to the healthcare population in South Africa. Just talk about that a little bit and the reasoning behind that, and then the strategy of how to address that.
So I think it's important just to place it into context, in that the South African strategy for rollout of COVID-19 vaccines was looking at 3 different phases. In phase 1, it was meant to be healthcare workers. In phase 2, it's meant to be individuals at high risk of developing severe disease. And then in phase 3, the more general public. Now, when it comes to healthcare workers, we need to appreciate that the South African healthcare working force, although there is a percentage of them, probably about 15 to 20%, that are at high risk of developing severe disease, the other 80% probably are not at high risk of developing severe disease. They are at high risk of being infected to the virus because of them working in healthcare facilities, which is sort of completely overwhelmed with individuals with COVID. So they're at high risk of being infected, but not necessarily all of them being at high risk of developing severe disease. So in the context of a vaccine, which we now show is not actually efficacious in terms of protecting against mild or moderate infection due to the variant that is dominating in South Africa, it wouldn't be a wise use of limited resources for us to go all out and vaccinate all healthcare workers with the AstraZeneca vaccine, knowing that close on to 80% of them don't stand to benefit in any way, because they are the ones that even if infected would only develop mild or perhaps some moderate illness, which the vaccine simply is not protecting against. But that being said, it doesn't mean that the vaccine won't necessarily protect against severe disease. And I think there's good biological reasons to believe that even the AstraZeneca vaccine that hasn't been efficacious in preventing mild and moderate illness might still protect against severe disease. And the basis of that is that the AstraZeneca vaccine uses almost an identical technology as the Johnson & Johnson vaccine, in that they both are non-replicating vector-based vaccines, in addition to which, when looking at immune responses of these 2 vaccines in a healthy population, they're pretty much identical after the first dose. And we know that a J&J vaccine in South Africa, when evaluated in a population that were at high risk of developing severe disease, in fact reduced moderate to severe disease by about 57%, and severe disease and death by as high as 89%. So we got 2 vaccines that are built on similar sort of technology, as well as are inducing similar sort of immune responses, but they've been tested in very different populations, and they've come up with very different answers because of the different populations they've been tested in, as well as they've looked at different outcomes. as the main endpoints of the study. So the decision not to go ahead with AstraZeneca vaccine in healthcare workers, I think it's a correct decision, because, like I said, it would very much be a waste of vaccines to be targeting a group where they're simply not going to benefit much, at least for those individuals that don't have underlying comorbidities or not of above 60 years of age and at risk of severe disease. Rather, the vaccine should be repurposed to the more general population who otherwise wouldn't have access to vaccine, but still remain at high risk of developing severe disease.
Let's turn a little bit more deeper into these mutations, because people get confused when you talk about the UK mutation, the Brazilian mutation, the South African mutation. What is really, really, really more important is that these multiple mutations happening in the same virus, meaning the 501Y, the 484, and now the Californian 452. These numbers, by the way, are the locations of the amino acids on the virus. And you sort of put in this word here that there are now mutations, while we're focusing on where these two things stick to the receptor. The mutations on the other side around this virus that are occurring that are as important, it appears, in terms of absence of neutralization. You call this N-terminal domain and S1 and S2, highly technical. We've discovered that, we've looked at that. So share with me and with the audience, when we talk about these multiple mutations, it's not just a single mutation, and how more dangerous they become when it's just more than one.
Yeah, so absolutely. And I think the experiments that have been done in the laboratory attest to what you are mentioning, and that it is the constellation of mutations that makes the virus less likely to be susceptible or more resistant to the antibody that's been induced either by natural infection or by vaccine-induced antibody. So the experiments that have been done is that when we construct these viruses and only include one mutation, there's still some amount of activity of the antibody against the virus. When you increase the number of mutations to 3 mutations, depending on which component of the virus you're looking at, then you got even further diminished in terms of antibody activity. And then when you include a full set of mutations in these virus constructs and you test it in the laboratory, then you get pretty much complete knockout of antibody activity for some of the vaccines. So it's not just a single mutation that's important, it's really the constellation of mutations which cumulatively makes the virus eventually more resistant to both vaccine-induced antibody as well as to natural infection-induced antibody. So I think this is, again, a phenomenon that probably evolves over time. I don't think all of these mutations necessarily have taken place concurrently. What we're observing in the United Kingdom right now, where they've got what is known as a Kent variant, which basically evolved around about October as well. More recently, they've now identified that in that Kent variant, there's sort of a new a mutation taking place involving this what we call the E484 mutation, which is considered to be one of the more important mutations, rendering the virus to become somewhat resistant to the antibody that's induced by past infection from the original virus or the vaccine. So you are correct. It's really a constellation of mutations that are sort of a determining factor in just terms of how resistant the virus becomes to the antibody that has been induced.
So, and you're seeing these constellations of mutations now in South Africa, as I understand it, when we talk about the 484, 501, and in fact, what was presented, there were 8 mutations, and now you're seeing even more in the same virus.
That's correct. And there are some critical mutations, at least 5 of which are directly associated with the type of antibody that we're wanting to work against the virus. So that's what's rendering some of these vaccines not to be too active. In fact, for some of them, they're not active at all, at least when it comes in relation to the antibody that's being induced. There might still be other underlying immunity that's been induced through vaccination, what is known as T lymphocyte immunity, which is somewhat being relatively conserved compared to the activity of the antibody that's been induced. But again, it's really the constellation of these mutations. And in the South African variant, there's at least 5 mutations, which are especially important, which is making the virus resistant to the antibody that's been induced from past infection, as well as, unfortunately, as an example, with the AstraZeneca vaccine.
And I think the final subject I want to cover with you, Professor Glenda Gray also presented the results on the Johnson & Johnson vaccine, which was a single-shot vaccine. Can you comment a little bit about that in terms of how you're going to approach that single-shot vaccine in South Africa?
Yeah, so I think the Johnson & Johnson vaccine was evaluated as a single shot under emergency use and in response to resurgences. It doesn't necessarily mean that a vaccine is going to be sustainable in terms of its protection or after a short period of time. And I think that sort of results still need to be forthcoming in terms of the duration of protection that's induced by a single dose. I would be very surprised, considering the immune responses that are induced by a single dose, that you're going to get sustained protection. I think even with a Johnson & Johnson vaccine, under emergency conditions, when you're faced with a resurgence, it's great that you can get protection after a single dose. In fact, even with the AstraZeneca vaccine, before the variant arose in South Africa, when we analyzed the data on a post-hoc basis, we found that even 14 days after a single dose of the AstraZeneca vaccine, it was able to reduce infection, mild and moderate infection from the original virus by 75%. So I think all vaccines, including the messenger RNA vaccines, which induce a very sort of somewhat mediocre immune response after a single dose, are still getting protection against the original virus that was circulating. But it's really the duration of protection which is important. And I think with the Johnson & Johnson vaccine, we probably would require a booster dose, either with a Johnson & Johnson vaccine or perhaps with another vaccine. So a combination of vaccines might well lend itself to even a more robust immune response and a broader repertoire of immune response than giving the same vaccine twice over. So the Johnson & Johnson data clearly provides hope in that when you're experiencing a massive resurgence, get in with the vaccine, give as many people a single dose as quickly as possible, and you can protect people from severe disease and from dying, or at least substantially reduce their risk. But as a long-term strategy, I think for most vaccines, it's really going to need to be a prime and boost strategy, either with the same vaccine, but probably preferably not with the same vaccine, because you do get some level of immune tolerance when you're using a similar sort of construct, and you might be able to overcome that to some extent if you were to sort of combine vaccines, which is where the field is really heading to, even in terms of the evaluation of the first generation of COVID vaccines.
So that was an interesting concept. Was now you say the field is now heading towards combining vaccines, a Johnson & Johnson vaccine with the Pfizer vaccine, or Johnson & Johnson vaccine with the AstraZeneca vaccine, etc. And I think it's a potentially good approach because perhaps these antibodies, all antibody-based, by the way, S and S and S, would have maybe a synergistic effect. We don't know that. I question, however, the logic of taking a sort of relatively ineffective S vaccine and a relatively ineffective another S vaccine and putting them together and hoping you get a better S vaccine. And a better logic would be to say take an S vaccine with an N vaccine, and now you have activation of two parts of the immune system. And again, with full disclosure, that's what I'll be working with you and the country. So before we end this session, our viewers always want and should have not only real hope, but hope based on science. So if you could leave our audience with some words of hope and inspiration of where you see this going. And then frankly, if there are any other things that you want to share with us that I've not covered, I'd love you to have that opportunity.
Thanks, Patrick. And I think after the disappointment which I've experienced in the past few days with the results of the AstraZeneca vaccine, clearly there needs to be reason for hope. And I think the hope does still exist. I think there's a shining ray of light that is upon us in terms of there being optimism that what we can do immediately is protect people from dying from COVID-19. We can substantially reduce their risk. We can safeguard our healthcare services. We can assist our healthcare workers by being able to vaccinate people and reduce the chances of being able to develop moderate disease as well as severe disease, even with the vaccines such as the AstraZeneca vaccine. So I think we're in a very different space than we were 6 months ago when there was very little hope, in fact, There was a lot of optimism, a lot of speak, but nothing really on the table which showed that the first generation of COVID-19 vaccines were going to work at all. In fact, even the regulatory authorities had already set a threshold of 50% efficacy to warrant a vaccine moving forward to being used under emergency utilization. So we basically have reached that sort of threshold, and the current vaccines have worked fantastically against the original variants that were circulating, but currently we do experience problems in terms of the evolution of the virus and the ability of those vaccines to continue. But the summary of it is we've got vaccines, we've got life-saving vaccines, and we cannot dismiss the current generation of vaccines, but at the same time, we need to be exploring better vaccines that are more suited to purpose, not just to protect against severe disease, but that are also able to reduce the transmission of the virus, that are also able to protect against asymptomatic infection, or at least reduce the infectiousness of people that are asymptomatic and are able to reduce mild infections. And it is with that type of vaccine that we will hopefully be able to get back to some level of normalcy in terms of our life.
Well, I can't tell you how much I want to thank you, not only for your time, but for your dedication to this work. And I really look forward to coming back home again to continue this conversation.
Thank you, Patrick. And we look forward to welcoming you back home, and we look forward to working with you. So thanks for hosting me.